Dear students, today we will have the second session, second part of the environmental biotechnology, that is biotechnological methods of pollution management. Last class, we saw how biotechnological methods are used for detection of pollution. And in this class, we'll be seeing how these biotechnological methods are used for pollution management or pollution mitigation. Let us see the class. So, the development of biotechnology has made huge changes in all facets of biological sciences, or in fact, in all facets of uh, social living. And here, in the case of environmental biotechnology, its development has resulted in the effective development of very effective methods for pollution abatement or mitigation. And we'll see in what all aspects these environmental biotechnology are used. The first and the most important and the most common one is the sewage treatment. And we know that sewage is the wastewater resulting from various human activities, including domestic, agriculture, and then industrial activities. Sewage is not such, when we say sewage, we usually think of human sewage or fecal sewage. It's not fecal sewage by itself. So, any domestic, agricultural, and industrial waste can be termed, as a result of human activities, can be termed as sewage. And you know that sewage is most commonly composed of organic and inorganic compounds, and it contains several toxic substances, even heavy metals, and several pathological organisms. And during the processing of this biology, we cannot dump this sewage as such into natural resources. Okay, we need to treat this sewage, make it harmless. Okay, so here, this for treatment of this biological sewage, this organic material is subjected to biodegradation or simply bacterial oxidation. Okay, so here the organic material is degraded. Organic material that is present in the sewage is degraded into smaller molecules like carbon dioxide, ammonia, phosphate, etc. Okay, so we know that here oxidation takes place, bacterial oxidation takes place. What is oxidation? Oxidation is addition of oxygen. Okay, so for biodegradation to take place, constant supply of oxygen is required, and this usually in the traditional process of sewage treatment, this continuous supply of oxygen is provided by bubbling with atmospheric oxygen. That is using filters and all we bubble oxygen into the treatment plant. And this is a very expensive process and requires a huge amount of manpower. So, what is an alternative for this? So, the alternative for this is uh, growing microalgae in the treatment plant. Okay, so by growing microalgae, continuous supply of oxygen can be achieved. How? Because we know that algae are very efficient in photosynthesis and they can release oxygen into the form in the form of dissolved oxygen. So, instead of expensive manpower requiring constant supply of oxygen or bubbling of oxygen, by growing microalgae, we can provide natural oxygen in the field and thereby speed up the process of biodegradation of sewage. Okay, and there is an algal bacterial symbiosis. We know that bacterial oxidation naturally takes place in sewage. Okay, so when we provide algal culture into it, so algal bacterial symbiosis takes place and a speedy, quick biodegradation of organic sewage matter takes place. And you know this is a natural and relatively inexpensive process. And second usage of biotechnology in pollution abatement is the use of bioscavengers, especially to heavy metals. Okay, and know that heavy metal pollution is a very drastic thing. And most of the heavy metals like lead, cadmium, mercury are all toxic chemicals which cause so much of problems to living organisms, including man. And there are two aspects of heavy metal pollution. One is bioaccumulation, and the other is biomagnification. Okay, bioaccumulation is that when an organism is exposed to heavy metals, it can accumulate the heavy metals in its body. Okay, upon constant exposure, these organisms can accumulate heavy metals in their body, and that is toxic. and this is called bioaccumulation. At the same time, when we know that fish accumulates heavy metals when it is grown in a, or when it is caught from a polluted environment. Okay, and this contains when the higher consumer, that including man, eats that fish, the same amount of heavy metals that is present in fish may get transferred to human beings also. Okay, but at the same level, at the same concentration, more toxic effect will be produced on human beings or higher consumer. You know, so there's a progressive increase in the concentration of in organisms at successive higher trophic level. At each higher trophic level, this concentration of these heavy metals increases, and this is called biomagnification. The concentration of the toxic heavy material present in the lower trophic level organism, when it reaches the higher trophic level, the concentration increases and the toxicity also increases. And this is called biomagnification. So both bioaccumulation and biomagnification is related to heavy metals. So in this context, it's very important to remove the toxic materials from the environment. And it's a difficult task. Okay, and there are that removal, even detection of heavy metals in environment is a very expensive process, and we can use certain bioscavengers. What are scavengers? Scavengers are those organisms which can clean up the environment. Okay, so bioscavengers like phytoplankton, some benthic aquatic plants, microorganisms, etc., can take up the heavy metals from the water and act as natural bioscavengers. And several plants can be used as bioscavengers for metal pollution, and examples are water hyacinth, then water lettuce, ferns. These all can take up heavy metals into its body and clear up the environmental heavy metal pollution. And examples are algae such as chlorella can take up heavy metals like copper, mercury, and uranium, and some microalgae like alga and laminaria. They are also used for the abatement of metal pollutants. Some fungi like Rhizopus, Aspergillus, Penicillium, Neurospora. These are all good absorbers of heavy metals like lead and mercury. So these are bioscavengers. Some bacterial species like E. coli can take up mercury, and Bacillus can take up copper from in a polluted environment. So these um, act as bioscavengers and very much help the natural recovery, natural abatement of heavy metal pollution from the environment. So these are the functions of bioscavengers. Then about eutrophication. Then what is eutrophication? That when sewage or other waste materials are being dumped or flowed upon into the aquatic environment, what happens? These sewage and wastewaters are very rich in organic and inorganic nutrients. So there's a huge flush of nutrients there. Okay, and these nutrients contain mainly phosphorus and nitrogen, and this will result in the huge growth of plankton and microalgae there. Okay, and these nutrients favor excessive growth of algae, and ultimately this results in the oxygen depletion. Okay, and thereby animals cannot get sufficient dissolved oxygen for their life, and this phenomenon is called eutrophication. So when sewage and wastewaters are influxed into the natural aquatic bodies, there is a huge flux of nutrients there, that is mainly phosphorus and nitrogen, and these nutrients favor excessive growth of algae, which results in oxygen depletion, that is called eutrophication. And this eutrophication results in the death of those organisms which cannot resist this pollution. Okay, mainly these are fishes, and this results in the huge mortality of fishes. See, there are huge amount of phytoplankton bloom there, and that results in eutrophication, and result is the death of fishes like this. This is only fish only, but usually huge amount of fishes are being dead there. Okay. And there are chemical methods available, such as the use of algae killing chemicals like algicides available for the control of eutrophication. But the problem is that these are not specific algicides. And when we use these algicides, the overall health of the complete ecosystem is lost. We cannot control. We cannot control or we cannot target this specific alkicide to that specific alkyl only. Okay, so there is just like beating the bush. So, chemical methods are always at the risk of overall health of the ecosystem. At this context only, this application of biotechnology comes. Okay, we can use biological approaches. For example, cyanophages, blue-green algae. Cyanophages um, can kill. Um, the cyanophages or like viruses can kill algal cells, which can be used to control eutrophication. Okay, so the usage of say this is how when you streak the virus cyanophages here, usually there is full of growth there, and later when we this is algal culture. See, this is algal culture, and when we use cyanophages here, see the reduction in the turbidity. Okay, so cyanophages can be used to control eutrophication. Okay, this is only at lab level. When we use it at natural level, more will be more result will be achieved. So this can be used to control. Cyanophages can be controlled to used to control eutrophication. And then another one is the usage of immobilized cells in sewage treatment. And the cells immobilized on certain surfaces are usually utilized for treatment of sewage. For example, bacteria like Pseudomonas butida and Cryptococcus elinovi are used for removing phenol, specifically phenol from the sewage. Phenol is a toxic chemical that is present in sewage. So, to remove phenol, Pseudomonas bacterial species can be used, and also Arthrobacter species can be used to triethyl lead, very toxic. Triethyl lead can be removed using Arthrobacter bacterial species. Copper can be removed by immobilized fungus Rhizopus. So this is Rhizopus. So this type of cells, microorganisms, can be used for the but immobilized cells can be used for sewage treatment. Okay, and this can be also used for the recovery of metals from industrial effluents. Okay, when industrial effluents are being dumped into the water body, what happens is that we can recover the essential materials, and this can be reused. And so these are very much useful in the pollution abatement. And then there is very importantly bioremediation. So what is bioremediation? It's the process of using living microorganisms to clean up a contaminated site. To clean up a polluted site, we use living microorganisms as such in the site. Okay, that is called bioremediation. And microorganisms do this by removing toxins or pollutants from the materials, and usually they do this by do this decomposition by the help of different enzymes that are present. And most of the times, single type of organism, microorganism, can completely regrade a pollutant, but some other times a consortium of bacteria, many bacteria are needed to for the complete degradation of a particular pollutant. Okay, and for example, we know that so much of water bodies are being contaminated with petroleum products, oil products. So for degrading, bacteria can be used for degrading the petroleum products which are being spilled into the water bodies. For example, the bacteria used for petroleum degradation are Pseudomonas, Flavobacterium, Arthrobacter, and Acetobacter. Okay, and the major advantage of these bioremedial agents like bacteria is that these can do this degradation work at the site itself, at the polluted site itself. Okay, and thereby we can avoid the need for physical removal and cleaning up of waste. Okay, but if it is not that, we have to first collect the contaminated from water from that we have, then we have to treat. But these bacteria can be act or bioremediate at the site itself. We can just spray this bacterial culture into this contaminated site. They can work on it. Okay. And for bioremediation, we can use both natural as well as genetically modified organisms. Okay, both can be used for bioremediation, and know that for treating soil and groundwater contaminated for a variety of chemicals, we can use these or recombinant and natural microorganisms. And one great example here is naturally occurring bacteria called Rhodococcus. Okay, and this Rhodococcus, this is the Rhodococcus culture, and this Rhodococcus can act upon the uh, pollutants in different pathways. For example, um, they can survive in solvents such as ethanol, butanol, then toluene, and they can kill, they should kill many other bacteria. Okay, so this rhodococcus is resistant to several chemical substances like ethanol and butanol, and there this principle can be used to degrade these chemicals, these pollutants, okay, by using rhodococcus culture. And there are several oil degrading strains of rhodococcus which can adhere to oil droplets. And uh, the importance of rhodococcus is that these are found in all types of environments at all regions, all geographical regions, like waste sediments, tropical soil, Arctic soil. So it's a cosmopolitan distribution. It's got a cosmopolitan distribution, and this rhodococcus can be very well used for pollution abatement. And a very important example for use of technology, that means a genetically modified organism using for pollution abatement, is Pseudomonas putida. And this is a genetically engineered organism, and this work was done by an Indian American scientist, that's Anand Chakravarti. And this was during the Gulf War period. This bacterial strain, was developed during the Gulf Wars time, just to but mitigate the water pollution that was caused by the oil spills. And this bacterium, this Pseudomonas putida, could utilize hydrocarbons, petroleum products in oil spills as carbon sources, thereby rapidly degrading the pollutants. Okay, and this genetically modified organism was named superbug. This is called superbug, and this was the first genetically modified organism to be granted a patent. This is Anand Chakravarti with the patent certificate. So, this Pseudomonas putida was genetically modified so that 5 or 6 contents of petroleum can be or oil can be degraded at the same time. Okay, so this biotechnologically modified Pseudomonas is called superbug, and this was a patented organism, and it was created by Dr. Anand Chakravarti, and this was the first GMO or genetically modified organism to be granted with a patent. And this example shows that naturally occurring organisms, so Pseudomonas putida is naturally occurring bacteria. This can be used as such for after modification by genetic engineering to process both biological as well as chemical waste. And um, what is the role of biotechnology in biodegradation? So the biological transformation of organic compounds by living organisms can be done using microorganisms, and we know that most of the pollutants contain xenobiotics. That means unnatural and synthetic chemicals such as pesticides, herbicides, weedicides, etc., and also other organic compounds. So these xenobiotics are very difficult to be removed, and if at all it is possible, it's very expensive. Okay, so biodegradation of xenobiotics is cost effective, and so biodegradation is the what immediate need for degrading xenobiotics. And what we said earlier, the Pseudomonas is a genus of bacterium that can degrade a wide variety of xenobiotics that we saw earlier. So, the Pseudomonas species is capable of degrading organic compounds like hydrocarbons, phenols, and then biphenyls, aromatics. These are all can be degraded using Pseudomonas bacteria. And these Pseudomonas, when it is genetically modified, it can be more effective in biodegradation process. So here, a particular strain of microorganism may degrade one or more compounds. Okay, even a single organism, single bacterium can degrade one or more compounds, but most of the cases, a consortium, a group of 2 or more strains or species may be more efficient than a single species because more amount of pollutants can be degraded at the same time. For example, the insecticide parathion is more efficiently degraded by the combined action of Pseudomonas aeruginosa and Stalseri. Okay, so this when two bacteria are combined together, the more efficient degradation of pollutants can be done. And there are several factors which affect biodegradation. One is the chemical nature of xenobiotic, how this xenobiotic is chemically made up, and also the capability of the individual microorganism that we use for biodegradation, the nutrient and oxygen supply, temperature, pH, all chemical, physical-chemical parameters will affect that. And usually, when we see the chemical features, generally aliphatic compounds are more easily degraded than aromatic ones. And then the presence of cyclic ring structures uh, then decrease the efficiency of biodegradation. Water-soluble compounds may be more easily degraded, and the presence of halogens in aromatic compounds inhibits degradation. So the chemical structure of the pollutants is the main factor that affect biodegradation. Also, the molecular orientation of organic, we said that in water science, this chemical nature of xenobiotic very much affect the speed of biodegradation. And then two terms we need to know. That's one is biostimulation. Biostimulation is just the cleaning up of the environmental contamination. And here, what we do is that we release certain nutrients or oxidants or even electron donors into the environment, which can stimulate the naturally occurring microorganisms to degrade the chemical. Microorganisms are already there in water. What we do is that we release certain nutrients or certain antioxidants or electron donors into the environment, which can stimulate these microorganisms to speed up the degradation of the contaminant. Okay, this is biostimulation. Another term is bioaugmentation. Bioaugmentation is a new approach. Here, along with the we actually add specific microorganisms into the contamination site. And along with this microorganism, we put the energy sources so that these microorganisms can multiply. Okay, so the microorganisms we add into the site. At the same time, we supplement that with the energy sources at the contamination site with a view to degrade the contaminant. Okay, so the introduced microorganisms may be either genetically engineered. If it is genetically engineered, the process will be more effective. Okay, so this is biostimulation and bioaugmentation. So these are the different methodologies we use for biotechnological methodologies we use for pollution mitigation or abatement. So two things we studied. One is how biotechnology is used for detection of pollution, and second one is how biotechnological measures can be used to mitigate or slow down or abate the pollution. Okay. So thank you very much, dear students. And if you have any doubt, please come back. Thank you.